Great. Hello and welcome to this class on the cellular automata and the models of artificial life. I'm Barbara. I will be giving the lectures. I'm a postdoc in the group of Clément Hongler, and we do research on topics related to this course. And together with me there's Esan and Jonas, who will help with the exercises. So the course in its current shape or form is completely new at EPFL, and as far as I know, it's not taught in this format anywhere in the world. It might be taught, or it will be taught next semester at Harvard by my colleague, but it's not even sure it will be taught next year at EPFL, so it's quite a special moment in time. So it's gonna be really about the theory of artificial life, and most of the lectures will just be Blackboard lectures, but not the first one, where my aim is just to give you an overview of what the field can be about, and well, the main purpose is just to get you excited. So let's try to make it, I think this should be fine. So let's take a step back and appreciate the fact that some 4 billion years ago, when the Earth looked something like this, some of the simplest forms of life emerged at the ocean. Yes? Ah, perfect. Thank you. One more thing is that because of the ephemerality of this course, it's being recorded, and well, you're not captured by the cameras. But I would love if you can ask questions during, and it might just be the case that those will be recorded, if you're okay with that. Right, so the simplest forms of life emerged in the ocean and managed to diversify in this fascinating process, leading all the way to us being in this classroom and thinking about this. And yeah, this is really one of the most fascinating questions on what exactly, or how exactly did life emerge, that remains to be open. There is actually a whole series of questions that are at most partially resolved. So yeah, how did life really emerge on Earth? And what is even life? So if you look into a biological textbook, there is a standard definition based on a list of properties that every living organism should have. That usually entails properties like homeostasis, an organization of the organism. It should have a metabolism, it should be able to grow and adapt, response to stimuli, and reproduce. But the problem is that it so seems that all the living creatures that we are aware of came from the same common ancestor. So it is not really clear whether this is the only solution to life, or whether this is just the solution that was once, or the properties that were once kind of invented by this common ancestor, and then we just happen to preserve them. And it is not clear whether these are really, whether there could be other forms of life that don't have these properties. So I will just put it side to side with a definition by NASA, which is more aimed to, let's say, studying life in extraterrestrial environment. And there it's a bit more reduced and more general. So, okay, it says it's a chemical system, but other than that, it just says that it should be self-propagating and undergoing adaptive evolution. So the self-propagation, in order for it to exist and maintain itself, it has to have some form of self-propagation, and to distinguish it from some uninteresting forms of self-propagation, like crystal growth, there is some notion of being able to adapt and maybe grow in complexity over time. But it is unclear, maybe there are other definitions that could be based solely on some information theoretic definitions, like life being able to reduce the entropy around itself. So this is definitely, in its abstract sense, we don't have a good definition of life yet. So if we just took a snapshot in time right now, without any prior context, it would probably be quite surprising to find something as complicated as us in the universe. But somehow the fact that we were able to evolve from simple creatures and that those were able to self-replicate creates like this probabilistic loophole, which makes the probability of us existing higher. But how did nature even manage to solve this paradox of self-replication? So if we look at all the examples of machines that humans created, it's usually the case that a machine can only create a machine that's simpler than itself. And it seems quite natural. Even if you take something like a 3D printer, it can print its parts, but it's unlikely that it was going to print a completely fully functional new 3D printer that's going to start printing right away. And indeed, it seems like if a machine A was supposed to build a machine B, then machine A has to have complete information about B. And on top of it, it has to have some mechanisms that are able to interpret that information. So it seems like A has to be inherently more complex than B. So how is it possible that we are able to self-replicate? So this question is actually partially resolved. And when it comes to the way information is stored about us in this inherently continuous world, at least on the scale that we perceive things, how come it's stored as discrete symbols? Then to illustrate two more questions about self-organization. So in the context of morphology, how can complex biological form arise reliably from local interactions among cells with no central blueprint or controller? And a related question, maybe, is what mechanisms make an ant colonies or flocks of birds self-organize without any central leader? And finally, what are the key factors that drive evolution to be truly open-ended? Because it really seems like there's no a priori upper bound on the complexity that there could be. And these questions are extremely fascinating, and they are actively studied by a variety of fields, but that will not be the purpose we will not delve into this particular research. We will aim to study or think about these questions on a more abstract level. And I would just like to highlight some of the key words, which is emergence, life, self-replication, complexity, self-organization, and evolution, that we can kind of think about in abstract terms. And yeah, I think these questions are as exciting as they can be, and everyone should think about them. And it is the case that a lot of people did think about them, not just in the context of these fields, but from a very theoretical perspective. So I will now mention three very exciting examples of some of the sharpest minds of the 20th century that did think about these questions and made some very interesting contributions on the side of getting Nobel Prizes or saving the world during the Second World War. Yeah. So the first one we will mention is Erwin Schrödinger, who got a Nobel Prize for his contribution to quantum mechanics. But also 11 years after that, he published a little book. He calls it a little book. I wouldn't consider it very little. What is life? Where he aimed to understand the physical aspects of life, of living cells. And one question he asked in particular is, what are the properties of the material that should store information, like hereditary information, in the cell? And just for the context at the time, this was in 1944, it was known that there are chromosomes and that they have to do something with hereditary properties, but it was unclear what exactly are the molecules that store that information and what are the key mechanisms or its key role in reproduction. And he did not really ask this question in an exact chemical setup. He was not aiming to find the exact molecule. It was more of a conceptual question of like, what kind of properties should that information carrier have? And he identified two factors. Well, it should be stable enough to just survive all the thermodynamical fluctuations, but it should also be rich enough in structure to be able to encode all various rich information about us. And actually, he was the first person who coined the term aperiodic crystal, which was exactly the type of property that he was describing. So the crystal part was the resilient solid part that should withstand the noise, and the aperiodic should refers to a structure being able to take enough forms to represent rich information. And just an interesting historical window is that in 1953, when there was a huge breakthrough in discovering the structure of DNA, actually, after their first paper, in August of 53, Watson and Crick sent a letter to Schrödinger, which I don't know if you can read, so I'm going to read it. Dear Professor Schrödinger, Watson and I were once discussing how we came to enter the field of molecular biology, and we discovered that we had both been influenced by your little book, What is Life? We thought you might be interested in the enclosed reprints. You will see that it looks as though your term aperiodic crystal is going to be a very apt one. Yours sincerely, Francis Crick. So, it's the first interesting or fascinating line of work. The second example that we will cover in detail in this course comes from John von Neumann. He was a mathematician with an extremely wide range of contributions, which include mathematical logic, quantum mechanics. He built the first fully digital computers, was a part of the Manhattan Project. He contributed greatly to game theory. And amongst other things, in the 1940s, he got interested in the relationship between living systems and computational systems, specifically between the nervous system and complex computing systems consisting of many parts. And he drew very interesting parallels between the two, which led him to a very fascinating question of what is the logical organization of self-replication and how can we build machines that can self-reproduce and overcome the paradox that we have talked about earlier. And he came up with a very important conceptual distinction which helped him solve this seeming paradox, and that's to decouple the organism into two parts: a tape, which contains information, for instance, descriptions of machines, and a machine which can act upon the tape. And if this was to reproduce as a whole, he would kind of consider some abstract models of physics, which would consist of some elementary particles out of which both the machine and the tape can be built. So we can think of them as some embodied Turing machines. And essentially, he argued that if the machine which acts on the tape is complex enough that it can interpret a rich range of descriptions that it's put on its tape, then if we put on the tape the description of the machine itself, which would literally just say, okay, first grab this elementary particle from the space and merge it with this particle from the space and continue, then the machine would actually build an exact copy of the machine. And the visionary idea was then to use the description that is on the machine's tape again, and this time to be copied onto the tape of the new machine, which really completes the construction and allows the new machine to start self-replicating again. And this double usage of the information is really the exact same principle that biology follows. It's the translation and transcription. And again, fascinatingly, he first talked about this idea in 1949, so 4 years prior to the discovery of the DNA structure. Except for this time, Watson and Crick did not send him a letter because at the time they did not make the connection of von Neumann's contribution to theoretical biology this connection was only discovered a posteriori by another Nobel laureate, Sidney Brenner. We will watch a video where he recounts for this fascinating part of science history later in the course. And so initially, the model which von Neumann considered to really construct a self-replicating machine was the kinematic model. And within this model, he already illustrated the core of his ideas. But there were a lot of technical details. Like the kinematic model really consisted of a three-dimensional continuous space, which would have some basic particles floating around that could be assembled by manipulated by other particles and so on. And just dealing with the mechanics and the motion of such particles in the space really distracted from the main purpose, which was the logic of self-replication. So in order to really complete his construction with full mathematical rigor, he actually switched to another model that was suggested to him by Stanislav Ulam. So just out of curiosity, who here knows about cellular automata? Great. So just a very brief description. A cellular automaton, in this case a two-dimensional one, consists of a potentially infinite grid of cells, or an actually infinite grid of cells, depicted here in gray. And each cell is assigned a state from a finite set of states. And each cell is also equipped with the exact same neighborhood. So in this case, von Neumann considered an immediate neighborhood where each cell sees its left and right neighbor and its bottom and top neighbor. And lastly, the cellular automaton is specified by a local rule, which essentially tells it if a particular cell's neighborhood is in this configuration of states, then the central state should change in this way in the next time step. And this local rule is applied in parallel synchronously to all the cells, which gives rise to global dynamics, which is often very complex and visually fascinating. And von Neumann designed exactly such an automaton in 2 dimensions with 29 states. You can see a particular configuration of a part of this infinite grid, which represents some signal transmissions. We will talk about this later. But indeed, in this environment, he was able to build exactly the tape and exactly machine of enough complexity to be able to actually build its own copy, and that copy also being able to create another copy and so on. It did take a while before this self-replicator was actually able to be implemented, because the original construction consisted of tens of thousands of cells, so simulating it at the time was clearly infeasible. So let's watch an example of such an implementation. So here you see a part of the cellular grid with the square configuration being the machine and this long line being just a part of its tape. I think in the actual world, the tape would come somewhere around here. The description is very long. And now we have seen the translation where the machine builds the new machine, and now we're witnessing the transcription. So the new tape is being built and information is being copied on it. So this actually is the bottleneck time-wise, but it should be done around now, and now you can see the new machine starting to build another copy. The third example of a very interesting contribution that we will also cover towards the end of the course comes from Alan Turing, that I think I need not introduce. He was a mathematician and essentially a founder of the field of theoretical computer science, who also helped to solve the Enigma cipher. But in 1952, he actually published a paper called The Chemical Basis of Morphogenesis. And there he was interested in a very fascinating problem. So how is it possible that an embryonic cell, which essentially starts with the chemical concentrations inside it, or with its overall structure being almost perfectly symmetrical. And how is this symmetry able to be broken in such a way that it always reliably leads to the complex pattern that it's somehow encoding, such as a horse, which is not a perfectly symmetrical organism. So here you see the initial phase of the symmetry breaking. And astonishingly, he was able to explain a part of this phenomenon by a very abstract model called the reaction diffusion model that he suggested for this. So just to quickly explain it, actually this kind of reactions is not what he considered. In his original paper, he considered linear ones, which were much more amenable to a rigorous analysis. These ones will be called the Great Scott equations, and they still miss some terms, so this is solely for illustratory purposes. But let's imagine the following simple scenario. Let's imagine there are two cells, and within each cell there are two chemicals. We can call them U and V, and they're specified by their concentration in cell 1 and in cell 2. This can be written by small u and small v. So this could be for cell 1, for instance. And they react according to the two following laws. So the first law is diffusion, which is just that if the concentration of U is larger in cell 1 than in cell 2, then it's going to flow from cell 1 to cell 2. And the second one, the reaction part, comes from a particular reaction process, for instance, one described here. And it's just kind of governs how locally the concentration of the two chemicals interact in each cell. And interestingly enough, he showed that since these equations are symmetrical with respect to the two cells, if one starts with two concentrations that are exactly the same in the two cells, then they will stay the same over time. This is the symmetry. But if one perturbs the system slightly from this equilibrium, the equilibrium is actually not stable and the system is being driven away from it. So this explains the symmetry breaking modeled by these reaction diffusion systems. And Turing considered more interesting topologies than just two cells. And even with just in the 50s, he already predicted that if there's like multiple of these cells having interesting topologies, it can lead to the emergence of fascinating patterns. And indeed, since we can simulate it now, here is, let's say, a two-dimensional grid of such cells, which starts with an almost perfectly symmetrical initial concentration. So this is the model of the Great Scott equations. And fascinatingly, these patterns are actually very similar to ones present on the skins of animals. And they are actually called the Turing patterns and are a crucial point of study in theoretical biology, thanks to Turing. So this summarizes the three examples where mathematics was able to say very non-trivial, interesting things about biology. And I think this is the true motivation for artificial life. So let's summarize what the field is about. So I think the main aim of artificial life is to study the fundamental mechanisms of life, abstracted away from the particular details of its biological implementation on Earth. Or yet another motto is that one studies life not as it is, but as it could be. And a central feature is that we try to recreate aspects of life in simulation. And these aspects are deeply related to the notions that we discussed before, such as emergence, self-replication, self-organization, evolution, and complexity. And the central problem is that these notions are still not rooted in mathematical definitions. But luckily, ALife in the modern days has progressed greatly by producing a wide range of fascinating examples which help us elucidate parts of these notions. And I think it is the central aim now should be to really root these examples in theory and help us elucidate what do the examples show and what do they not show. Just to briefly go over some of them. Maybe this is the reason why you signed up for the course. I would just be interested who's here because they find the game of life interesting. Nice. So yeah, an iconic example comes from the 1970s, and it's a cellular automaton with very compact rules. It's again on a two-dimensional grid with just a very limited neighborhood where each cell can be either dead or alive. Yet when we initiate the system randomly and just visualize the dynamics, it produces quite fascinating unexpected patterns. And in some sense, game of life does seem to be open-ended in the following particular sense. Well, it is proven to be Turing complete, and people do love to use game of life to actually program in it and construct patterns of fascinating complexities that can do interesting things. For instance, this is a pattern that essentially computes Fermat primes. So to this day, it's a hypothesis whether there exists a Fermat prime larger than 65,537. And someone designed this configuration such that it would annihilate itself if and only if such a prime exists. So this is the open-endedness of game of life. And to push the compactness even further, there is rule 110, which is a one-dimensional cellular automaton, so now only acting on a one-dimensional grid of cells. Again, each cell can be just dead or alive and has an immediate neighbor to the left and right. So now you can really specify the local rule just by 8 bits of information. Yet this rule was proven to be Turing complete. This is how it looks from a random initial configuration. So here every row is a particular configuration and time is progressing downwards. So this is the space-time diagram produced. So it does have some structures that travel in space. But it is actually an extremely elaborate proof that helped to show that this rule is indeed Turing complete, and it requires a very particular encoding of the information into the space-time diagrams and a particular way of zooming out to see the computation. So here you see how the computation looks like. There are some vertical static gliders that represent information or data on the tape, and these are being manipulated by other sorts of gliders that react with it. So there have been various constructions of self-replicators in cellular automata with ranging properties, some of them very compact ones, such as this loop, which is able to self-replicate and on top of it essentially perform an arbitrary computation based on the little tape that it has attached vertically. Some constructions even allow for certain sorts of inheritable mutations, such as these evoloops, which upon interacting with each other can change each other's genome, and that manifests phenotypically in loops of different shapes. So typically evolving towards the very small ones that replicate fast. So that's also quite interesting. And actually this leads us to some very complicated designs of systems which are typically initiated with the first generation of self-replicators that interact with each other and evolve all sorts of properties. So these are examples we will not cover in the course, but still they could be quite fun to look at for you, such as the bibids, which are these organisms which have tiny brains represented by small neural networks, and they actually find food and digest it. And interestingly, in some sense, they do satisfy the whole list of properties that a living system has that we have seen at the beginning. So it's still a question, is this system live then? And another interesting system with self-replication and emergent phenomena is the Alien project, again consisting of elementary particles that interact and aggregate to form these creatures, which can be parasites or hosts, and create their own ecosystems. So I really recommend you to explore this. So this leads us to continuous models of cellular automata that we will also touch upon. So the space of continuous systems is in some sense much easier to navigate because now we can use continuous optimization methods to search for behavior that we would like to see. And this is indeed what happened not so long ago. And some neural cellular automata have been trained to model some aspects of morphology. So essentially here, the neural cellular automaton again has a local neighborhood, but now each cell can be in a state from an infinite continuum of states, for instance, encoding the RGB colors. And this neural cellular automata encode the trajectory it takes from a small embryonic cell to grow such a complex shape and somehow maintain its structure even to pretty large perturbations sometimes. Or it's also used to generate all sorts of fascinating, even dynamic patterns. So this one has actually been trained by Ehsan, your TA. So they also look pretty cool. And lastly, another version of continuous cellular automata that we will briefly look at is Lenia, which can be in some sense interpreted as a continuous version of game of life. Which again, we can see the emergence of gliders with various properties and even multiple of them interacting. And we will also cover in more detail the reaction diffusion models that we have seen before. So great, so just to summarize, what this course will aim to do is to present the main theoretical results relevant to artificial life and hopefully put some of the examples you have just seen in the context of this theory. And we will specifically focus on three aspects of dynamical systems. It's their dynamical properties, their capability to compute, and their capability to self-replicate. And we'll try to present some results of how these properties relate to each other. So an example of some questions that we will be able to at least partially answer is, what are the abstract mechanisms of self-replication? What is the relationship between self-replication and computation? What does it mean for a dynamical system to compute? How to search for systems with complex behavior in some automated manner? And what is the role of dimensionality in all of this? And finally, what this course does not aim to do is to teach you biochemistry or molecular biology. It also does not aim to blindly mimic biological life without elucidating any general mechanism behind it. So a great example of this that makes it into some textbooks about the field, but is a bit misleading, is the 18th century construction of a mechanical duck. And lastly, what this course will not aim to is to get you a job at Google. So unfortunately, the set of skills we will acquire here, I don't think translate well to industry, and that should not be the reason why you're here. It should be, I suppose, the good reason to be here is that you just find the questions we have talked about fascinating.